microsporogenesis, and megasporogenesis. So any one of you have any confusion in microsporogenesis, megasporogenesis? What is microsporangium? What is megasporangium? And anything related to it? Any name, any word, any part? Now is the time to speak. So we did both the microspore and the megaspore. Anyone has any doubts?
No, sir.
Okay. And also, I think related to Iram's question, Iram asked one question. I saw this pollen grain structure and it came to my mind. So Iram was pointing out at one time that in one of her notes, it is somewhere mentioned that the tapetal cells form the exine. Is that what you said, Iram?
Yes, sir.
Yeah, so that's not true. Okay, I think we discussed this, and I hope that was clear to you. So, what is a pollen grain? A pollen grain itself is just two cells, right? At some stages, it is a three-celled. So it is just two or three cells, correct? And exine is nothing like a cellular structure. You can think of exine as something like a cell wall. So how is cell wall formed of plants? If you think about a cell wall, what is a cell wall? Is it a cell? It's not a cell, right? It is a it is a multi-layered structure. Okay, Parker, if you want to quickly rejoin, you can do that. Maybe that might solve the issues that you are having with the mic, and I'll wait for you. Okay, if you want that. Okay, cool. Yeah. Okay, so we have a good news that Iram is sharing, and I don't know why you people write it in the chats. You know, it makes it's okay. I can go back and forth to read it. But Iram has something to share with us. Iram, would you like to speak now? It's a good thing.
No, sir. I will not.
No? You would not like to share it with your batchmates?
No, that's just kind of like showing off.
And that is?
So that's just like showing off.
That's not showing off. Come on. If you don't mind, then I can share with your batchmates.
Yeah, if you want to.
Okay, cool. So let Parker join, then we'll talk about it. But yes, so regarding your question, since it was your doubt, what I wanted to say is just like a protective layer, a cell wall, which is a multimeric layer of, in case of plants, it is made up of cellulose. So we call it cellulosic cell wall, right? In case of bacteria, it's peptidoglycan and other things. In case of fungi, it's chitin. Similarly, in case of pollens, that protective layer is sporopollenin. So chitin is also a organic molecule, which is secreted by the same fungus that gets deposited over it and makes that layer. Similarly, the tapetal cells do secrete sporopollenin, but the cells itself cannot form the exine because otherwise to cover two cells, you would not like to put more cells around it. Makes no sense, right? And so do you understand how is sporopollenin making the exine? And sporopollenin is not the only thing that's present in the exine. There are a lot of other chemicals present in the exine, but sporopollenin is what give exine its characteristic strength and feature, that it's almost indestructible. Okay. Chemically. Does that make sense, Iram? So
yes.
If anywhere they say that tapetal cell makes or forms the exine, I assume that the conversation is in context of that glandular tapetal cells secrete sporopollen, and hence they help in formation of enzyme. The layer itself is not cellular. Yeah, so it just occurred to me. So yeah, so that was microspore. Any doubts in megasporangium? So in today's class, I will keep going topic by topic because most of the topics that are left, we have discussed it already. I will rediscuss or dictate things that are important for exams in today's class, and we'll try to finish this chapter. So you have to tell me if you have any doubt in megasporangium, ovules, its structure, all these labels that you can see on your board. Any doubts, Rabia, No?
So,
Yes, Kiran.
so there was a numerical from this chapter that came in my exam.
Okay.
Yeah, it came in my exam, this exam term one, and I did some mistakes. So,
Would
will
you
I
like
share
to,
that question here?
yes,
Yeah.
right. It will be great if
It's
you
a long
share that
question.
question. Yeah. So we will discuss that question. Great. Just share the question in the chat and we'll discuss it. Okay. By the time Iram is sharing the question, Rabia, do
So
you
it's a
have any
long
doubts?
question.
Yeah. So like, do you have a problem sharing it in the chat or?
So I have like the paper with me. I have the hard copy. So how am I supposed to share the chat?
Okay.
It's long actually.
Yeah. Right. And we cannot share images. Can we can share images in the chat, right? Can you see an option to share images on the chat? Are you using computer or mobile phone or what? If you're using a mobile, can you see that? Can you see any option? More. Emoji format. Type a message here. Can you share any image? Or you can do one thing. Great idea, Iram. You are on the WhatsApp group, right? That we have.
Yes. Yeah.
Yeah, just share that. Just share a photo of the question there. I'll pull it out from there.
Okay, sure.
Okay.
Sir.
Yeah.
Just give me a second.
Yes, and Parker has also joined in. Parker, can you hear me? And is your mic issue resolved? Doesn't look like the mic issue resolved, but I hope you can hear me. Okay, great. If you can hear me, that's great. So since Parker has also joined in, so Iram has a good news to share with us, which she is hesitating to share herself, but she wrote in the chat that I'm very happy to share it with you that Iram has scored 67 out of 70 in her bio term 1 exam. So that's very good. I think Iram deserves, yes, Rabia is already clapping. And I really appreciate. Great, Iram. I don't know how can I clap? How are people clapping? Sorry, I'm like,
Sir, it's in the reactions.
yeah, I'm a little more. Where are the reactions? Sorry. With me.
So,
Yeah.
so do you want to know where I lost marks?
In
Very
this.
silly mistake. This one and one more question was there, which was, write the alternative name for MTP. That is induced abortion, right? I wrote
That was
induced.
MTP is yes. MTP is medical termination of pregnancy. That is induced abortion, chemically induced abortion.
Yeah, they've asked the question for this, and I wrote induced pregnancy. So
MTP. Okay. So remember the word T is for termination. Okay. Induced pregnancy is mostly these are the ART techniques, the assisted reproductive techniques, where you do either ZIFT, GIFT, IVF. All these are to induce a pregnancy, but to terminate a pregnancy, that is MTP, medical termination of pregnancy. If it was induction, there must be a word I, not T. Just some, but yeah, that was a silly mistake indeed. But it's okay if you are doing silly mistakes. That can be forgiven, because silly mistakes can happen by anyone. Even I used to commit a lot of mistakes. But yes, that's a great score, 67 out of 70, and keep it up. Okay. Okay, so Iram has shared the question number 22, right? This is it. Let me pull it out, paste it here. Okay, let's see if I can put it here. And it will be great if I don't know why it's not pasting, but wait. Yeah, now I can do it. It says JPG, but it's not coming up. Can you see the question? No, right?
No,
No.
sir.
Yes or no?
No, sir.
Maybe now you can. Yes. Can you see the question now?
Yes, sir.
Make it. Okay, so since this question is shared by Iram, Rabia, would you like to read it? Let's see if only you can solve it, and then I'm not required at all. Yes, would you like to read question 22?
Okay, sir. A flower of brinjal plant following the process of sexual reproduction produces 360 viable seeds. Answer the
Mhm.
following questions, giving reasons.
Mhm.
A. How many ovules are minimally involved? B. What is the minimum number of pollen grains that must land on stigma for pollination? C. How many
Mhm.
microspore mother cells or pollen mother cells must have undergone reduction division prior to de- dehiscence of anther in the above case?
Yeah. So let's break it down. Okay. Let's come to details, and let's also see if these details are any help, or they're just, so sometimes in questions details are just to confuse you. Okay, you have to first learn the art to figure out whether these details are even necessary or not. So a flower, which is a sexual reproducing organ of a brinjal plant. So what do we know about brinjal plant first? So here comes the knowledge. What kind of a flower or what kind of fruit brinjal is? Let's start with that. Rabia, what do you think? Brinjal is true fruit or false fruit? What do you think? So brinjal, I told you that all the vegetables are basically fruits. We call them vegetables. So for its own plant, brinjal is a fruit, right? And brinjal belongs to the plant Solanaceae, about which you must have studied in class 11th, where you studied the floral formula of. So Solanaceae was one family, Liliaceae, Solanaceae. Remember these families?
Yes, sir.
Yeah. So brinjal, this kind, and you will be if you don't know this, you will find it interesting. Brinjal belongs to the berry family. So it's a berry. The fruit is a berry. The eggplant is a berry. Okay. And what happens in brinjal? How many seeds do you see in one brinjal? Multiple seeds, right? Brinjal plant have multiple seeds, yes or no?
Yes, sir.
Yes.
Do you all have internet with you at the moment? Of course, like, and if you have any
Yes,
other
sir.
device, just search brinjal and look at an image. Can you see a brinjal? Basically also known as aubergine or eggplant. But just look at an image and tell me, where do you see the seed? So you can write cut brinjal or something, sliced brinjal, to actually look at the seeds. Where are the seeds? So if I cut brinjal, and this is the cross section of brinjal, okay, where do you see the seeds located? You will see the seeds located in some or the other arrangement like this, right? Do you see this?
Yes, sir.
Yeah. This is how seeds in brinjal are located, correct?
Yes, sir.
Now it looks like there are multiple compartments, yes or no? And you are right if you think that they are compartments, and within that there were multiple ovules. So it is a multi-carpellary flower. So it produces 360 viable seed, it says. So one brinjal plant, so when it says a flower of brinjal plant produces 360 viable seeds, when it is producing the seed, it has become brinjal fruit by now. Okay, it's not just the flower. To produce seeds, it has to undergo pollination, and seed is the result of post-fertilization. The ovules become seeds. So when I say seeds, you directly understand ovules. So now it's clear that there were 360 ovules that got matured into seeds. Okay. Now you have to answer the question, how many ovules are minimally involved? It's very, very simple answer. How many ovules were involved? There are 360 seeds. 360, isn't it? There could be, why they are asking minimally involved? Because it's possible that there were more than 360 ovules in that process, in that flower, but all of them did not mature into seed. Some just degenerated or could not become seed or could not get fertilized. But minimally, if there were 360 seeds, there has to be 360 ovules that got, because ovules are future seeds and ovaries are future fruits. So it's a very simple question if you think about it. What do you think, Iram? Do you understand, everyone? So minimally, there has to be 360 ovules. Otherwise, how will you make 360 seeds? One ovule, one seed. Make sense?
Yes, sir.
Yes.
Yeah. Then what is the minimum number of pollen grains that must land on stigma for pollination? Yeah. How many each seed has one embryo? Always remember that. So if each seed has one embryo, there are 360 seeds. So how many embryos? How many
360.
embryos? 360 embryos. And how is the embryo formed? When an egg cell fertilizes with fuses with a male nucleus, correct? So how many male nucleus must be present or are required to make 360 embryos? Simple, 360. You need 360 male embryos to fuse with 360 egg cells to make 360 embryos. Sorry, male nucleus, I should have said. Did I say male embryos? It should be male
Yes,
nucleus. So
sir.
we need 360 male nucleus to fuse with 360 egg cells present in the egg in the chamber, what do we call it? The sac, and hence 360 embryos will be formed, which will be present in each seed. So each seed, when they say the word viable, it is a viable means it can germinate. And this is a hint that it contains embryo inside it. Okay. If they say that there were 60 viable seeds and 20 non-viable seeds, so in that case, you do not need a male nucleus. It's unviable because there are other ways to make seeds, but then it's not a fertilized seed. It cannot germinate because it does not have an embryo. It only has endosperm. Okay. Or maybe it also does not have that. So viable is a very important factor, that it has live embryo inside it. So we need 360 minimum number of pollen grains that should land on the stigma. So this was very, very straightforward, but just they're asking you in a confusing manner. Is that clear, Iram and Parker and Rabia?
Yes, sir.
Yeah,
Yes.
Iram? Now the question C is where the actual trick lies. How many microspore mother cells or PMCs must have undergone reduction division prior to dehiscence of anther in the above case? So remember, we just need 360 to make the above case happen. And they use the word microspore mother cell. If they would have used macrospore mother cell, remember, one macrospore mother cell divides into a tetrad, but only one out of the 4 develops into a ovule, right? But in case of microspore mother cells, one microspore mother cell, which is called PMC, pollen mother cell. So one PMC divides meiotically to form a tetrad, and each cell of the tetrad becomes a pollen grain. So in this case, you have 360 pollen grains. So you will do 360 divided by 4. And the answer is, how many microspore mother cells must have undergone reductional division?
90.
Exactly. So for this case, the answer is 90. Any doubt? Any confusion?
Sir, what about
Just
mega spore mother cell? If they
yeah,
were
if
asked this question.
3, then the answer would be, what would be the answer then? What do you think?
360.
How many? Yes, you need 360 megaspore mother cells to undergo reduction division, because every megaspore mother cell, when will undergo reductional division, will form a tetrad. But the problem here is 3 will degenerate, only 1 will form a ovule. So to make 360 ovules, you will have to make 360 megaspore mother cells undergo the whole process of megasporogenesis. So basically, they are asking the question from megasporogenesis. In this case, they are asking from microsporogenesis. Is that clear?
Yes, sir.
Perfect. So thanks for sharing, and I hope you understood the logic. So just pay attention to what they're asking. First, if they would have asked about mango plant, then this question would not have been this tricky, because we know that mango plant has just one seed. But for brinjal and all other berries, which actually are true fruits, but have multi-carpillary structure, there they can have multiple true seeds. And to make multiple true seeds, you need multiple fertilizations happening, and so on. Okay, now let me go back to where I was asking you things. So all of you are clear with megasporogenesis and microsporogenesis? Any doubt in that? No? So let
No,
me
sir.
ask Rabia a question. Rabia, what is a megaspore dyad? I told you about megaspore tetrad, but what is a megaspore dyad? DYAD. That is a term in your NCRT. Any idea what is a dyad? Do not get intimidated by the question. The question is very simple. Just be confident and say that yes. What is a megaspore dyad? For reference, you can come to the figure 2.8 of your books. And you will see that there is a megaspore dyad mentioned. Iram, would you like to give the answer?
Yes, sir. When the MMC undergoes the first meiotic division, then it's a dyad, right?
First meiotic division. What do you mean by first meiotic division? It only undergoes one meiotic division.
There are two, right?
There are two what? There are two meiotic divisions. At the end of one meiotic cycle, there are 4 daughter cells produced. So at the end of 2 meiotic cycles, there should be 8. So what you are trying to answer, I know that you are trying to give the right answer, but you're not putting it in a correct manner. What you want to say is a dyad is formed after meiosis 1 completes and meiosis 2 has yet not completed, right? So one cycle of meiosis has 2 subcycles, meiosis 1 and meiosis 2. And you have studied in your previous classes that meiosis 1 is the real reductional part. Meiosis 2 is just like mitosis only. Meiosis 2 mimics mitosis. Yes?
Yes, sir.
Yeah. So a dyad is the intermediate stage where the megaspore mother cell has just completed meiosis 1 and has 2 daughter cells, which are both are haploid. And because they are 2, it's called a dyad. Now it will complete meiosis 2 also. And finally, there'll be a megaspore tetrad. Is that clear, Rabia? Oh, Rabia is not here. Is that clear, Parker and Hiram? Give me a thumbs up if that part is clear to you. Okay, perfect. Pollination is clear to both of you. Any doubts in pollination?
No, sir.
No? Okay. Pollinators is also done. Okay, so there is one paragraph in your book which called outbreeding devices. Okay, I would like, this is an important paragraph. Iram, can you open that paragraph in your book, Iram and Parker, both? I think it's
Yes, sir.
somewhere around, yeah,
Page number.
after, yeah, I think, I'm not sure what page number it is, but there is a paragraph called
31.
outbreeding. 31. Okay, thanks. Great. Parker, can you open page 31 and come to outbreeding devices? I think it's a small paragraph that's there. Did you find it, this paragraph? Apparently, there are questions that come in boards from this paragraph. And they ask the questions. You can twist the question to make it look easy, difficult, and also add or subtract components from it. So Iram, would you please read this paragraph for us? And please stop wherever I say stop and we'll discuss what it means, okay? And
Yeah,
read
yeah,
it a
sure.
little slow so that we can understand. Yeah, outbreeding devices.
Oh, yes. A majority of flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flower.
Right.
Continued self-pollination result in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and encourage cross-pollination.
Yes, this is something we were discussing in the last class. Remember, both of you?
Yes.
Yeah, continue.
In some species, pollen release and stigma receptivity are not synchronized. Either the
Yes, that,
pollen is...
yeah, just stop. So this is one of the tactics that I dictated you. The question can come, like you have already written the question, under what scenario a plant can favor or disfavor one type of pollination over the other. So when plants encourage cross pollination, the one way to do is to desynchronize the release of pollen and receptivity of stigma. That is point one. Then continue, Iram. Just for Rabia, Rabia just joined in. Rabia, Iram is reading a paragraph that's labeled as outbreeding devices in your NCERT, and it is on page number, what page number, Iram?
31.
31. Okay, Rabia, can you hear me?
Yes, sir.
Yeah, come to page number 31 and Iram is reading that paragraph. Yeah, continue.
Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of pollen.
Yeah,
In some
it is
other
just explaining
species,
the first thing only. Yeah.
yes.
So this
In
is
some
one
other
type.
species,
Sorry, continue.
yes. In some other species, the anther and the stigma are placed at different positions so that pollen cannot come in contact with the stigma of the same flower. Both these
Yes, now stop
devices
here.
prevent.
No, just stop here. Stop here. So this sentence, it is just a one sentence, but what it is pointing towards is whether the ovary is placed superior or inferior, whether the stigma is taller or shorter than the ovary itself, something that we were discussing in the last class, this part, right? Remember this?
Yes, sir.
Yeah, so this line which is saying that in some other species, the anther and stigma are placed at different positions. So if the anther is smaller than the stigma, the pollen cannot land here on its own. So this is to discourage self-pollination and encourage cross-pollination. Okay, this is strategy number 2. Okay, by making the ovary superior or inferior to the anther. Yes, continue.
Both these devices prevent autogamy. The third
Yes,
device to prevent inbreeding is self-incompatibility. This is a
right.
genetic mechanism.
This is an important part for which I asked you to read. So all of you just make a short note on self-incompatibility. This is a genetic mechanism. It has molecular reasons and genetic reasons behind it. Basically, you must have got the hint from the name itself. Whatever happens, the plant, even if its own pollen falls on its own stigma, there won't be a pollen tube formation and there won't be a fertilization event happening by the self pollen. So there is something that helps the plant to know that this is my self pollen and this is the pollen from some other plant, but of my own species only. Okay, so the species remains same, and it can identify between self and non-self, just like animal cells can do, right? So remember, something similar to self-incompatibility I have taught you in terms of humans. Graft rejection, remember that? One human donates a kidney to another human, it's a human kidney, but the cells, the body's immune system can understand whether these cells belong to my own body or not and can show a response against it. That's called self-incompatibility for the tissue. Does it make sense, everyone? Can you connect dots, the parallels in biology?
Yes, sir.
Yeah, they do not happen by the same mechanism. Remember that, but just to give you a insight. Yes, continue. What is self-incompatibility? Read it.
This is a genetic mechanism and prevents self-pollen from the same flower or the other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil.
Yes. So the pollen from the same flower or from other flowers from the same plant. So basically, this is a genetic mechanism to prevent autogamy and geitonogamy both. So this is a question that has come in entrances from this part and also in boards. The question is, how can a plant avoid both autogamy and geitonogamy with some surety or with some confidence? So there you can say self-incompatibility. All plants do not have this. Let me tell you. Okay, all plants do not have this mechanism, but some species have evolved this mechanism. And these species are basically those species where variation matter more than just replication. Like me and Iram were discussing, we all were discussing in the last class. Iram asked the question that why would a plant not like to have variation, right? So we discussed that in environments where change is not happening fast, where there is no selection pressure as such, you might not see, mutations or variations as advantageous, right? Only when things change or there is a selection pressure, then variation is very, very advantageous, right? So these plants live in areas where selection pressure is there constantly. So they need variation. Now, how to ensure variation and also ensure fertilization? Then they go into self-incompatibility mode, that I will not allow my pollen to fertilize my flower. Okay. They don't want to rely on just superior or inferior ovaries, because still there are chances that same pollen can land. They also don't want to rely on, or maybe they cannot do very, very properly the desynchronization between pistil receptivity and pollen release. So they go one step ahead and develop a foolproof mechanism, which is a genetic mechanism. By genetic, I mean there are genes that play a role in it, and some proteins are released which do not allow the pollen pistil interaction to happen if the pollen is from the same plant, whether from the same flower or from a different flower, but from the same plant. Okay. And it will be important for those plants, as I said, that need variation to constantly keep surviving and flourishing. Yes, Iram, read.
Another device to prevent self-pollination is the production of unisexual flowers. If both male
Yes.
and female flowers are present on the same plant, such as castor and maize, monoecious, it prevents autogamy but not geitonogamy.
Deuteronogamy. Yes, just stop here. So three mechanisms we studied: desynchronization, okay, superior, a different arrangement, superior inferior ovary. The third is more foolproof, which is a genetic mechanism, self-incompatibility. Now they're talking about the fourth mechanism, a fourth solution to the problem, that you produce unisexual flowers. Now there is some bit of confusion sometimes. When I say unisexual flower, it's pretty straightforward. Uni means single, sexual flower means either the flower has a female system, which is pistil, or it has the male system, which is stamen, right? But if on the same plant, you have both. So one flower is male, and on the same plant, another flower is female. In this case, of course, autogamy won't happen, but geitonogamy, which is biologically autogamy only, correct? Is that clear, everyone? Geitonogamy is biologically autogamy only, right?
Yes.
You cannot prevent that, right? So what can you do? If you are a plant, you cannot desynchronize, you cannot do structural arrangement differently. You also cannot develop genetic incompatibility. But what you can do is you can make unisexual flowers. But if you make unisexual flowers, but on the same body of the plant, both the flowers are there, still geitonogamy can happen, and it is of no help. It will also not give any variation, because it's biologically self-pollination only. Just by process, it mimics like cross-pollination. So what can you do? What is other one step ahead of this? Is there any other way? Yes. Then they start behaving like how humans are. Then one plant becomes the male plant, it will only bear male flowers. And one plant becomes female plant, it will only bear female flowers. Does that make sense?
Yes, sir.
And this will for sure avoid self-pollination and only allow cross-pollination. So if a plant, a single plant, have both male and female flowers on it. I'm not talking about bisexual flowers. Bisexual, anyways, both are present in the same flower. But if there are two different kind of flowers, male and female, but they are present on the same plant, for example, castor and maize that they have given example, then those plants are called monoecious plants. The term, this is where students often get confused. So remember it for your entrances. The term mono, no, there is only one. Oh, monoecious. Now let's break it. The word mono means
Single.
one or single, yes. And the word oaceous comes from the word oesh, which means body, soma or body. So mono means one, oesh means body. So monoecious is one body. So in these species, the species have just one body. And on the same body, you will have both the sexual organs present. So monoecious plants have either bisexual flowers, that is one way to have both the sexes on the same body, or they can have both male and female flowers. Is that clear?
Yes.
So the moment I say bisexual, this plant has this species has bisexual flowers, what should come to your mind is that this species is monoecious, because both the sexes will be present on the same plant then. Okay. Now, if I say that a plant is dioecious, then Rabia, tell me, what does this means? Dioecious. If I say a plant is dioecious, what does it mean? If you have understood it. Rabia, are you there?
Yes, sir.
Yeah. Are you trying to formulate an answer?
Yes, sir.
Yeah, just go ahead. If you are wrong, we will try to push you to the right track. Let's start with dioecious. What does it mean? I am saying that this plant X, species X, is dioecious. What information do you understand about that species by this word, dioecious? Di means?
Two.
Two. Perfect. So very good to begin with. I think I'm getting some answers in the chat, maybe from Parker. Yes, Parker has given a right answer. Let's see. Rabia also reaches there. So Rabia says di means two, and oasis means
Body.
bodies. Okay. So what do you know about this plant now? If this plant is dioecious, then what does it mean? You just have to put two bodies in a sentence. It means that this species have two different bodies, right? If we consider human as plants, then human as a species is dioecious. Do you understand, Rabia?
Yes, sir.
Some books often use dioecious for animals, but this word is not that readily used for animals, it's used for plants. That's why I'm explaining this to you. So why do I say that humans are dioecious? Because humans, the species of human have two different kind of bodies. One we know as male body biologically, one we know as female body biologically, right? I'm not considering, or I'm not discussing here the other genders or biologically where there are anomalies and both the sexes are present in the same body, but just to make you understand. So if I'm saying that a plant is dioecious, it also means that this species of plant has two different bodies. In that case, one body will have male organ and the other body will have all the female organs. So in this case, what kind of flowers will be produced by dioecious? They will always produce monosexual flowers, right? Right? One sex on one body. Is that clear to all of you? Everyone give me a thumbs up if
So their
it's
names are
clear.
very contradicting to their flowers, right?
Exactly. Monoecious is bisexual, produces bisexual flower. You can remember it like that also. When we talk about flowers, then we talk about sex organs. And if in the name of flowers, there is the term bi or di, then in the body, there will be the term opposite, mono. If in the flower, there is the word mono, then in the body, there will be the word di. So monosexual flowers, dioecious body. Bisexual flowers, monoecious body. Make sense, everyone? Biologically, it makes sense, right? Because if you have a bisexual flower, you don't need two bodies in the species. You just need one body. Both the sexes are on that body only. Now, let me ask you a trick question which was asked once in board. Under what condition? Write down this question. Under any condition, is it possible? Under any condition or under what condition is it possible for a plant to produce monosexual flowers, for a plant to produce monosexual flowers and have monoecious bodies Who will give me the answer? Everyone wrote the question? Iram, Mariam, Rabia. Thumbs up if you have written the question. So this is where we are going in the details to make the concept clear. You know, one of my teachers, like my science guru in my life, always used to say that to cover the syllabus, first learn to uncover it properly. So go beneath the sheets of the syllabus. That's where the details lie, and that's where your fundamentals will become stronger. Everyone written the question? What is the answer to this question? Suppose this question comes in one of your exams or in boards. What will you write as an answer? You know? Are you there, Iram?
Yes, I'm thinking.
Yes, think about it. Monoecious flowers, monoecious body. Is it possible at all? If you think it is not possible, your answer has to be like, under no condition this is possible because this is contradictory to the fundamentals or something like that. If you think it is possible, then you have to tell me under what condition it is possible. And let me tell you, the hint is on the board right in front of your eyes. Rabia, if you would like to give the answer, please go ahead. Rabia? No? It's very simple a question, people. Iram, would you like to try or should I go ahead?
So please provide us with some hints.
So what did you say? Sorry, come again.
So please provide us with some hints.
Yeah. So the hint is, the question is simple. It is asking, can a plant have monosexual flowers, but still have a monoecious body in the species, for that species? Which means, can a plant have a separate male flower and a separate female flower, but not separate male and female bodies? Of course. That is the second category of being monoecious itself. Look at here. This is the hint. Can you see this? I just did not write it in the first place. Here, to be a monoecious, there are two possibilities. Either they have bisexual flowers, that makes them clear-cut monoecious, because bisexual flowers will have both the organs in the same flower, and then you don't need a different body. Or you have different flowers. So both male and female flowers, which means this is one monosexual flower, this is one monosexual flower, but you have both the flowers on the same plant. Then also, in that case also, you don't need another body. So it is possible for a plant to have monosexual flowers and have monoecious body. And under what condition? That given that both the flowers, both monosexual flowers are present on the same plant. Then the plant becomes monoecious as a species and also have monosexual flowers. Does it make sense, Iram and Parker? Was it very difficult?
No, so it was very easy.
It was very easy, right? So the
Mhm.
answers always lies in fundamentals. You just have to think, what are they saying? Now, if I ask you, is it possible for a plant to have bisexual flowers and a dioecious body? Under what circumstances is it possible? Is that possible? And if yes, under what condition? Can a plant have a bisexual flower and dioecious body? Iram, come on.
No, it's not possible.
It is not possible, because the moment you say bisexual flower, you don't need another body, because both the sexes are present in the same flower, and any plant that have that flower have both the sexes, so the body automatically becomes monoecious. So you cannot have a dioecious species which produces a bisexual flower, but you can have monoecious species that produce monosexual flowers. Write it down. This is how they trick you. In fact, there are plants like castor and maize. Castor and maize are monoecious species having monosexual flowers. Castor and maize have monoecious bodies and they have monosexual flowers. Okay, let's move forward from this thing. This was a little convoluted thing, so I thought we'll spend some time here. Pollen pistil interaction is clear to all of you, right?
Yes, sir.
Yeah. So the whole idea of pollen pistil interaction is that, let me ask you a question. Tell me true or false. Okay. So the question is, a successful pollination guarantees a fertilization event. True or false? A successful pollination guarantees a fertilization event in plants. True or false?
False.
False. Why? Why do you say so?
So it depends on the pollen grain. It doesn't depend upon the pollination. Because if you have
Yes.
pollen from a different plant, it won't work, right?
Right. Perfect. So that is the right answer, that it's false. Okay, Parker. Why it's false? Because even if the pollination is successful, the pollination only means transfer of pollen grains from the stamen to the stigma, either of the same plant or of different plants, same flower or different flowers. Okay, whatever, all combinations are possible. But pollination simply means the transfer. Transfer of pollen to the stigma does not guarantee fertilization, because if it is mango's pollen fallen on a guava's stigma, there will be no fertilization. Pollination happened. The pollinator first went to this plant and then came to another plant and successfully transferred the pollen. So pollination is successful. The pollinating agent did the work, but still there will be no fertilization, because the most crucial step is pollen pistil interaction. Is it clear, Parker? Give me a thumbs up if you understand this.
So,
Yes.
so how are there different varieties of mangoes?
How are there different varieties of mango?
Yeah.
Yeah, it's a good question. So different varieties or different breeds do not mean different species. All varieties of mango are a product of selective breeding. You select for that particular kind of traits. Do you know that all the dogs that you see, if I compare a German Shepherd with a bulldog and a bulldog with a Chihuahua and a Chihuahua with a pup, and a pup with a beagle, or a beagle with a Labrador, you might be thinking that all these are different species of dog, right?
Yes.
They are so different from each other. Imagine a German Shepherd and imagine a Chihuahua. But you're wrong. They are all the same species. Okay. Then how are they so different from each other? They have the same genes. They have the same number of genes. They can interbreed. The sperm of Chihuahua can fertilize a female German Shepherd. And there will be a progeny that will be a crossbreed between Chihuahua and German Shepherd. Okay. There might be severe problems and complications in how it looks and how it walks and things like that, but they are all same species. So that is called breeding. Breeding of dogs is a perfect example of what humans can do. That is called artificial selection. So that makes us believe in evolution. When anyone asks you that how are you sure that evolution happens? What is the proof of evolution? Evolution happens in a time frame of million of years. How do you know it even happens, or something like that is even possible? So one obvious evidence is fossil records. But let's say someone does not want to believe in fossil records. All these are man-made things. There is nothing like a dinosaur bone. These are all stories. Okay. But the next evidence that they cannot ignore is selective breeding. If we humans, without tempering the DNA, just on the basis of selective breeding, okay, so you breed one species, then you see, due to variation, one of the baby looks different. You select that and then you inbreed that. So due to selective breeding, we have been able to make multiple breeds of dogs which look very, very different from each other, morphologically, so different that one will not believe that they are same species. If humans can do it just by having the knowledge of genetics, in nature, where there is an actual selection pressure, a natural selection pressure to survive, don't you think this will happen? Of course, this will happen. So there, when it happens, that is called as speciation events. New species are formed. We are making new breeds because that can happen faster. Okay, making new species is a very long process, but breeds can be made. So all these, what are your question again? Sorry, I think I diluted your question. What was your question?
How are there different varieties of mangoes?
Yeah, varieties of mangoes. So they're also based on the morphology of what kind of fruit. So all the mangoes that mostly we get in the market are, they're called, what are they?
Alphantins.
No,
The
what is
name,
the
right?
scientific? Yeah, the scientific name.
Mangifera indica.
Mangifera indica. Mangifera indica is not native to India. Okay, Mangifera indica has been bred a lot in India, but it's basically, if I'm not wrong, native to Australia or somewhere. So Mangifera indica can have different varieties that we know as, Malda, Dasheri, XYZ, and so on. You can have, I think, a man made a world record of growing more than 100 varieties of mango on the same tree. So what he was doing is he was doing grafting of branches from different varieties of mango. And since their genome is same, they're all compatible to grow on the same plant. Okay. So it's selective breeding. So basically you put one type of mangoes, then you see that one of the tree have the mangoes which are little more sweeter and the mangoes look a little more reddish. Some change happened in their morphology or maybe in their genome, but it was very subtle change. Then you selected the seed of those mangoes and you started breeding them separately over generations. And then you successfully created one different variety of mango, which is separate from the parent variety with which you started. Do you understand that? That's how we make varieties. Yeah, we select for morphology, sweetness, pulp, and things like that. But they're all same species. Yes, that's a good question. So similarly, we do for rice also. All the rice are, I think, by some Oriza, but we have more than 50,000 varieties of rice in India alone. But we do not, some of these are genetically manipulated. High yielding varieties, HYV, some have vitamin in them. We have tweaked their genetics, but they're basically all same species. Same we have done for maize and other cereal plants. Okay, perfect. So the answer was false. That's pollination does not guarantee fertilization. Therefore, pollen pistil interaction is very important. And what happens in pollen pistil interaction? It is the ability. Would you like to write this? Write it down. It is the ability of the pistil. It is the ability of the pistil to recognize the pollen of the same species. To recognize the pollen of the same species through chemical interactions. Through chemical interactions between the pollen membrane, pollen cell membrane, and the cell surface receptors of pistil. Pollen cell membrane and cell surface receptors of pistil. Okay. We also discussed emasculation and bagging, right? Is emasculation and bagging clear to both of you, Iram and Parker?
So for emasculation, you said that we use a polythene or something to, bag it over, right? Over the plant, over the flower, right? But actually, it's written that you use butter paper for it.
For what?
For bagging.
Yeah, you can use butter paper also. So I see the idea is you have to use something that allows sunlight to come in. Okay. And also it does not have to be so airtight at the base where you are tying the knot. Some air should also come in, but pollen grain should not reach the stigma. Is that the whole point of bagging, right?
Yes.
Yeah. So you can use butter paper also. Fine. Butter paper or yeah. Not everyone who does emasculation have access to high quality butter paper. So I have also, normally you can use other things also which allow it. Like polythenes are the most basic thing, but I understand we should not promote usage of plastic in class. Yeah, so what is emasculation? You take a pair of forceps and then remove the anthers from the flower. And you have to do it before the flower gets mature. So mostly it is done at the bud stage when the bud is about to open, when it opens for the first time. And you know that it has not yet started releasing pollens. At that very moment, you remove the anthers. Okay. And yeah. And basically why we do emasculation, because we do not want self-pollination to happen, or we want to may fertilize them with a particular pollen of our choice. So if you want to fertilize it with a particular choice of pollen, then after emasculation, you also have to bag it. So bagging will prevent contamination from any other pollen, unwanted pollen, right? So only you can go and brush the stigma with the pollen of your choice or your variety, and then keep it bagged. So that's called emasculation and bagging. If you do bagging without emasculation, then you are preventing cross pollination and you are supporting self pollination, where the stigma and the stigma and the stamen are intact, but you have bagged the flower right from the bud stage. So bagging can be used for both, okay, to favor cross pollination and to favor self pollination. Make sense? Everyone, is it clear? Any confusion, any doubts?
Then at bud stage, you have the
Mhm.
anthers developed.
No, we don't have the anthers developed. That's what we want. If the anthers develop, like the structure will be there. When the bud will start opening, you will see that there are structures, right? But as I said, dehiscence will not happen. The PMC, the pollen mother cell, would just be in its initial stages of doing meiosis and making tetrads, and it will not have made fully functional pollen. We just cut at that stage. Yeah. So it won't be mature, but you don't want it to be mature.
It's very tiny, right? How are you able to cut it?
It's very tiny. Yeah, it's very tiny, and yeah, it's just. So let me tell you, I in the lab dissect out brains from fruit flies. So if you have seen how small is fruit fly, a bud is 100 times bigger than a fruit fly. So it is still very well possible. You just need a very small pincer or forceps or tweezers and just pluck it out. It's like plucking eyebrows. People do pluck eyebrows, right? It's possible to do. You can pluck eyebrows. You can pluck anthers from a bud. That's not that impossible a task, right? You know?
Yes, sir.
Yeah. But I understand what you're saying. Like it's not like anyone can go and then do emasculation. Yeah, you need some practice. True. Yeah, so that this process by which we are deciding what kind of breeding we will do, which pollen will fertilize with what stigma, this is called hybridization. So in nature also hybrids are formed. One kind of breed, you know, fertilizes with another kind of breed. That's called hybrids, right? But when it happens naturally, we call it natural hybridization. But when it happens through emasculation and bagging, then we call it artificial hybridization. So if the question comes, talk about artificial hybridization and the methods that is used to achieve artificial hybridization, you will talk about emasculation and bagging. Okay. Does that make sense? And Parker? Okay, perfect. Now we have to study in detail three things, which are the last three things. And I think before we go, these three things are endosperm, embryo, and there's one more thing. What is it? So the endosperm, embryo, and the seed itself. Okay. So these three things we will come at the very end. This will take one class. So the class that I have kept in reserve, which is the next class, I will use that to finish this. But before that, we can finish everything else. We also have discussed some aspects of these three also. So we know what is endosperm. Quickly tell me what is endosperm. Endosperm is a post-fertilization structure. Once fertilization has happened, and we know that in angiosperms, the fertilization event is called a double fertilization event. Why it is called a double fertilization event? Because there are two fertilizations that happen, right? What are those two fertilizations that happen? One male nucleus fuses with egg cell, that is first. This gives rise to an embryo. And the other male nucleus fuses with the polar nuclei, and it gives rise to endosperm, right? So
Yes.
endosperm is a triploid structure, a triploid tissue. It has 3N ploidy. Okay. So that is endosperm. And its major function is to store food and provide that to the embryo when the seed is germinating later on. An embryo is the result of male nucleus fusing with egg cell. It is diploid, 2N, and the development of embryo is called embryogeny. Okay. And there are two kinds of seeds that you must be knowing about. We'll go into the details in the next class, but let's finish all the basics. What are the two kinds of seeds that we know of? What is the difference between a
Monocot
rice?
and dicot.
Yes, monocot and dicot. Plants also, we can say that this is a monocot plant and this is a dicot plant. What is monocot and dicot means?
So in monocots, you have the endosperm. Endosperm is not consumed here. And in dicots, the endosperm is completely consumed.
Yeah, but how can I see and tell that the the endosperm is consumed or not consumed? Is there any other better way to, if we see, if we just go by the name? So what you are saying is correct, that in monocots, the endosperm is consumed by the embryo as it develops, right? And in dicots, it's not, correct? So you can see endosperm also in the seed. But the name monocot comes from monocotyledon, a single cotyledon. And the word dicot comes from the word di, two cotyledons. So any seed that you can naturally split into two halves is a dicot seed. It has two cotyledons filled with endosperm. And the example is all your pulses: moong, chana, tur, which is arhar. You took any peas, matar, right? You can just open the seed into two parts. It automatically just opens like a book, right? Like a pendant. Yes or no? Does it make sense what I'm saying?
Yes, sir. Yes.
So if you have seen a gram, a chana, it splits from the center when you eat it, right? And splits into two. Those are two cotyledons. That's why these plants are called dicotyledons, dicot plants. But the seed that cannot open, for example, rice. Have you ever seen a rice splitting into two easily? You can break it into two or three or whatever piece you want. That's a different thing, that we can break anything. But do you see a cotyledon opening in rice or in corn, maize? You all must have enjoyed popcorns. Can you split the corn seed into two from any excess?
No.
No, right? So corn, which is maize, rice, they're all monocots. They just have a single cotyledon, which is the seed, which forms the seed itself. Okay, so we'll come to the differences between these two kind of seeds in the next class, but you know what is monocot and dicot now. Okay, perfect. Let's also spend some time on the concept of roots. So yeah, go ahead.
So can you also tell about why orchids are an exception of dicots.
Why? Who?
Orchids.
Orchids, the flower.
Yes, it's an exception for dicots, right?
It is an exception for dicots. Yeah. So you want to know why, right?
Yes.
Yeah. So basically, orchids are monocots. We don't. The seeds of orchids are basically monocots, but they. To answer your question, why they are exceptions in dicot is because they do not have cotyledons. They lack two cotyledons is the first. This is a ground rule for all dicots. Dicots have cotyledons, and the seeds of orchid lacks cotyledon. Okay, that is first thing. So you can write down.
So under what criteria are they coming as an exception for dicots?
Under what exception for dicots? Why are they called dicots? Let me think about it. So I understand your question. They must be orchids are monocots, but they must be exhibiting some criteria because of which people often think that they're dicots, right? But we say that they are exceptions. Because I don't think they are. Is it the root? See, what I know about orchids is that they are monocots. They also are non-endospermic, like all monocots. Their endosperm is exhausted. So I don't know. How about we let me think about it, because
Sure, no
for
problem.
me, orchid. Sorry,
You can
tell
give
me.
the answer next. That's fine.
Yeah, because see, for me, I think orchid is clear-cut monocot. It lacks cotyledons. It lacks endosperm in its seeds. But why do people say it's an exception for dicots? I will think about it. Okay.
Yes, sir. Please take it.
Yeah. Maybe. Maybe something about their root structure and the flower symmetry. Maybe that is what I'm thinking at the moment. Okay, I'll get back to you on this. Okay, why they are
Yeah, sure.
exceptional dicots, but they're not dicots. Okay, even if they exhibit some feature of dicots that I will figure out and we'll discuss in the next class, but they are not at all dicots. Okay, they are
Yeah.
monocots, fully monocot. Yeah, yeah. At least all the orchids that I know of. Yeah, biology is full of exceptions. Okay, and orchids are very rarely, they're not very much studied plants. A lot of labs now are interested in orchids and how they propagate, but still those studies are limited. Yeah, so also what I wanted to quickly go about is, about from a seed, when an embryo develops, you all have done this germination experiments as a part of your summer holidays, I'm sure, where you take a gram, put it in the soil and it develops, right? Then you have to capture at different stages. Have you done it, or this is not in
Yeah,
fashion these days? Oh, you have done it.
we've
Great.
done it.
So yeah, then we click pictures and we say that what comes out of an embryo, there are two things, radicle and plumule. Do you remember that? The first two things that come out from the embryo, two extensions, are called radicle. One is radicle and one is plumule. Right? Plumule give rise to the shoot, all the green part which is above the soil. And radicle give rise to the root system, all the non-green part which is inside the soil. Right? Just brushing up. Correct? This is clear to all of you?
Yes, sir.
Yeah. So let's do one thing. And you go back and read about seed germination, radical, plumule, and something about dormancy. So you know that there is something called seed dormancy. What is a dormant period or seed dormancy?
It's an inactive period.
Yes, so dormancy means inactive period, dormant. But what happens in that inactive period? In that inactive period, the embryo, which is inside the seed, undergoes, like you said, dormancy, and it waits for favorable conditions. Okay. What is favorable conditions? Moisture around it, a particular temperature sometimes. That is what makes the seed start germinating. So you can store dormant seeds for any, like not for any, I should say, but every seed has a viability period that the seed will stay dormant. But if you do not put it under favorable conditions beyond this point or under this time limit, then they will become non-viable. So seeds stay viable. In that viability period, they may also stay dormant. So dormancy does not mean non-viability. It simply means that when favorable conditions are given, they will germinate. Okay, so just go back and brush up this concept, and in the next class, this chapter will be over. Okay, so we will focus on three things: embryo, endosperm, and seed. I think fruit we have done. I'll tell